All right, so welcome everyone. Today we kick off the El Zoominario series of 2021 with a bang with Claudia Solis-Lemus. She is now an assistant professor at the University of Wisconsin-Madison. She works at the Wisconsin Institute for Discovery and at the Department of Plant Pathology. Before that, she did a postdoc at Emory University. And before that, she did her PhD at UW-Madison in statistics, and she also did a master's in arts in mathematics. She did her undergrad in Mexico at ITAM in two majors, actually. One of them was actuarial sciences, and the other one was applied mathematics. She is now a world expert in network inference and phylogenetics. And today she's going to tell us about how to infer the tree of life. So Claudia, thank you very much. We're excited to have you, and please take it away.
Thank you, Daniel. And thank you all for joining us today. Yes, I'm going to be talking about learning the tree of life. And I wanted to start with this story that appeared in 2017. It feels like 100 years ago in the Washington Post, of an exhibition of dinosaurs at the Center of Science and Industry in Columbus, Ohio. In this exhibition, the peculiar thing was that some dinosaurs, like the T-Rex, were presented with feathers. And that's different from the idea that we have from T-Rex that we see in movies. So that was the peculiar thing on this exhibition. And then a couple of years later, there was another exhibition that was covered this time by the New York Times on the American Museum of Natural History, where again, we had the T-Rex presented with feathers. And I don't know about you, but for me, the idea of the T-Rex having feathers is a bit comical and even soothing, because the T-Rex is this vicious killer that appears in horror movies and is very wild and savage. But now picturing it with feathers, it almost feels like a giant chicken. So I don't know, for me, it has been quite funny when I heard these stories and the potential that these dinosaurs had, in fact, feathers covering part or the whole of this body. The truth is that we don't have enough scientific evidence to conclude that, in fact, the T-Rex had feathers or no. But one thing that we do know is that the modern birds that we see today actually evolved from the dinosaurs. So in this image, for example, I'm presenting that the dinosaur's limb, the limb of the dinosaur, evolved over millions of years into the modern day's wings of the birds. And this evolution process, of course, it happens over millions of years and is driven by random mutations of DNA. And some of these mutations are beneficial for the species, and of course, they get fixated. So perhaps because we know this connection between birds and dinosaurs, the idea of a feathered T-Rex is perhaps not completely far-fetched. You might be thinking at this point, well, wait a minute, how do people know this? How do scientists know that the birds actually evolved from lineage of the dinosaurs? And the answer to this question lies on the tree of life. The tree of life is this massive graphical structure that represents the evolutionary process from single-celled organisms at the origin of life into the diversity that we see today from plants, fungi, reptiles, birds. Everybody is in this tree. And to give you some intuition for those who have never heard of the tree of life, it is like a family tree where we have ancestors and descendants, but in the family tree, they're all human species. In the tree of life, we have all different living organisms displayed there. But this tree of life allows us to identify which species are closely related to each other. And we often use terms that are similar to family trees, like sister species, to define species that are related via a common ancestor closely into the present. So it is with the tree of life that we can answer questions about which is the organism that is most closely related to humans. In this case, it is the chimpanzee. Humans and chimpanzees are sister species because there was a previous ancestor from humans and chimpanzees that diverged into these two different lineages, humans on one side and then chimpanzees on the other. And it is also with the tree of life that we know that the mother birds evolved from the lineages of the dinosaurs. This lineage, of course, we all know it got extinct, but it was evolving into slowly, in this slow process, into the birds that we see today. So how do scientists know that the T-Rex is related to birds? Is via the tree of life. But then you might be asking, wait a minute, how do we get the tree of life in the first place? And that is actually the question that we're going to try to answer today. How do we learn the tree of life for different organisms? And I'm going to use the example, the toy example, with only 3 species, but you can generalize this to whatever number of species. So let's say that I want to get the tree for salamander, T-Rex, and birds. Now you're going to have to bear with me, because traditionally we do this tree reconstruction for living organisms, and the T-Rex, we know that it has been extinct for millions of years. So I'm just using it to follow up with the same example, but just keep in mind that we tend to do this tree reconstruction for organisms that are living and are extant today. So the question is, I have these 3 species and I want to get the tree that represents their evolutionary history. And one option could be this tree, where the colored nodes represent the external nodes represent the living species, and the gray nodes are the internal nodes that represent ancestral species. So for example, this gray node represents a species that lived millions of years in the past, that was the common ancestor for T-Rex and birds. And of course, then the other gray node represents the whole ancestor of the whole clade, the 3 species. This is one possibility of the tree. And if this is the tree that we reconstruct, we would say, yes, T-Rex and birds are closely related to each other. But another option could be a different tree. We could have the tree where actually salamander and T-Rex are closely related, and we would observe a node where there will be an ancestral species to both salamander and T-Rex. If this is the tree that we reconstruct, we would have a different conclusion. We would not be saying that the birds are closely related to T-Rex, but instead the salamanders. So there are, in fact, 3 possible trees that we can get from only 3 species. And we can see that if there are only 3 species, we either have T-Rex closely related to birds. This is the first case on the far left. Salamander closely related to T-Rex. This is the tree in the middle. Or the third option is salamander closely related to birds. That's the third option. So for us statisticians, it is the choice. We want to know which of these trees is the true tree, is the one that represents the true evolutionary relationship between these 3 organisms. So that is the question that we want to answer. And how do we do it? Well, we need to collect data. We are not going to guess randomly between the 3 options. We need to collect data and identify the tree that agrees with the data that we observe. And by data, I mean genetic information. So we would extract genetic sequences from the organisms in our study. And again, here, this is why we use living organisms, because we go out to the field and we grab the species and we sequence the data, and we cannot do that for the T-Rex. But that's what we would do. So we would go out there with sequence genes, and the way this genetic information looks like is just a string of 4 nucleotides. We know that DNA is comprised of the 4 nucleotides A, C, G, T, and therefore a gene is simply a string of these 4 combinations of these 4 letters. In this case, I'm showing again a toy example with a gene that only has 10 nucleotides, but in reality, we have genes that have hundreds or even thousands of nucleotides that we use to reconstruct the tree. So it is with this data that we collected that we want to infer which is the tree that represents the evolution of the organisms in our study. And the way that we do this is we need to have a mathematical function that quantifies how much agreement with data each of these trees have. So we will observe for the data that we have, which of these options is maximizing the agreement. And statistically, we have a name for this type of function. We call it a likelihood function. So the likelihood function is measuring how likely it is that we observe this data when we had the underlying tree as our true parameter. For example, if we just focus on the tree on the far left, if this is in fact the tree that maximizes the likelihood, it has the peak here, this would mean that this tree is maximizing the agreement with the observed data. And that's what we want. We want a tree that agrees with what we see from our collected samples. And then the question, of course, is if the likelihood function is pointing at the true tree, how do we calculate this likelihood function in the first place? And of course, this is a short talk, so I cannot get into all the messy details about the likelihood computation, but I'm just gonna show you what are the main ingredients of this recipe. And like I said, I'm showing you now an example that has 10 nucleotides. It's a gene with 10 nucleotides. But to simplify it, let's just start with just one nucleotide, which is one of those columns. So if this is the data that I collected is just one nucleotide where I observe A in the salamander, C in the T-Rex, and C in the birds, the first ingredient that I need to calculate the likelihood function is a probability model for the transitions. So if we can imagine that generations in the past, there has been some mutation process that causes some alterations in the genome, and the simplest of it is just a mutation in a site where there used to be an A in the descendant. Now there's a C because there was an error, because for whatever reason, now we observe a C. This is a mutation. And we model that with some probability model that tell us how likely it is to see each of these mutations. We can have a simplified model in which we assume all mutations are equally likely. So it is the same to mutate from C to A or C to T or C to G. They're all equally likely. That is a simpler model to a model in which we allow different mutations to have different rates. And all these parameters, they are just governing that, how likely it is to observe certain mutations in our data. And that is encoded in this Q matrix. The other ingredient that we need to calculate the likelihood are the branch lengths. Because of course, if we have not a lot of time for mutations to happen or get fixated, then perhaps we would expect not to see a change in that nucleotide. But if we have a longer time, if the branch is longer, then we perhaps expect a lot more mutations to accumulate in this longer time. So depending on the branch lengths, it's also how likely it is for the tree to produce the patterns that we see in the data. And the third ingredient is what we call the ancestral state. So we observe the nucleotide on the 3 species that we collected, but we do not know what was the nucleotide for the ancestral species that is not observed. So we need to put them as an unknown, X and Y in this case, but they also matter for the computation of the likelihood. So we can have we can estimate what could be the more likely ancestral state. Perhaps we can guess that Y was in fact a C because both descendants have a C, or it could be a different nucleotide depending on how likely these mutations happen too. So with these 3 ingredients, we can get the formula for the likelihood, and this is the only formula in my slides. The likelihood here denoted by capital L has these different arguments, has the tree topology, which is the shape of the tree, who is more closely related to whom. We have all the branch lengths, T1, T2, T3, T4. We have this ancestral states, X, Y, and we have the rate matrix that governs, like I said, the rate of different mutations. And this likelihood function is simply a product of probabilities. So I'm just gonna illustrate the first 2. So P of X simply represents the probability of observing X at the root or at the common ancestor of the whole clade. And then the second term represents the probability of mutating from X to A in time T1. So that's what this P represents. P of T1, that is time T1 of mutating from X to A. And all the other probabilities are the same. They're just transition probabilities, and we multiply them all to obtain the likelihood of this tree with these parameters. And of course, like I said, I'm not telling all the messy details. And for those who are familiar with probability model, this is a continuous type Markov model that we're using. But with that formula, all that we need to know right now is that it allows us to identify which one is the tree that maximizes the likelihood, and therefore it is the tree that maximizes the agreement with the observed data. And one thing that I had not mentioned is that, well, I focus on just one side for now, but the likelihood for the whole sequences is simply the product of the likelihood of each side. We assume that each side evolves independently. And now that sounds all very simple and easy, right? You just evaluate the likelihood on these three cases, choose the maximum, and that is your tree. It turns out that in real life, it's of course a lot more complicated, because the case with three species, like I said, there are only three possible trees. So it's very simple. We just evaluate the three options and that's it. But if our sample size increases, then the number of trees just grows out of control, to the point where if I have 52 species in my sample, I have more trees than atoms in the universe. So it is impossible to do the same pipeline. It's impossible to just evaluate all possible trees, because we will never end, right? So, and 52 does not even sound like a big number. The data sets nowadays are in the hundreds or even thousands of species. And we want to identify the tree that represents the evolutionary history. But of course, we no longer can simply evaluate the likelihood on all the possibilities. So things start to get a bit more challenging, because we have this high dimensional space, which is this tree space that is growing out of control. We need to optimize all the numerical parameters. So the trees, they're bigger, they have more branch lengths that we need to optimize. We need to find the maximum, but we, again, cannot evaluate all trees. So we need to have some heuristic search in this space and hope that we converge to the maximum. But like I said, this is a hope. Sometimes we don't have theoretical guarantees that we will converge to the maximum likelihood. And so, of course, this estimation process takes a lot of time and a lot of memory. So it is a very challenging problem to estimate this tree for larger data sets. And the other thing that makes things complicated is that there are challenges also in the terms of the model, the size of the model. For example, there are some species that hybridize. They create a hybrid species. And then the representation of the evolution can no longer be just a tree, because in the tree, when two lineages split, they become separate lineages and they just don't join back ever. But when there is a hybridization, you have two different lineages that they combine to create a new hybrid. And therefore, this joining pattern creates kind of like a loop that is no longer tree-like. So for certain organisms, the tree is not a good representation of the evolution, and thus we need to estimate phylogenetic networks, not phylogenetic trees. And in fact, the bulk of my research is devoted to producing methods to estimate phylogenetic networks from different types of data. And we produce this Julia package called PhyloNetworks, which has many options to estimate phylogenetic networks. And in fact, this is an open source tool. So if people are interested just to learn more about networks, or even learn about Julia, that I don't know if people have heard of Julia, this is a new programming language that is similar to R or Python, but it's faster and better in many senses. So if you're just interested to learn about Julia or to learn more about phylogenetic networks, you can just go to my website, and everything's on GitHub. So you can just look at the whole code and everything's available there. I want to conclude with just acknowledging the people that work with me, students that are really talented, and I'm really lucky that they decided to work with me, and a bunch of other collaborators here at UW and in other institutions. And now that was the end for my research part of the talk, and I'll be happy to take questions after this talk. I want to now talk a little bit about my culture, which is, well, I'm originally from Mexico, as you heard, and I thought it would be cool to tell you about the Dia de Reyes, which is something that we just celebrate on January 6th. So that was Wednesday, I think. And what we do, it's a very exciting day when you're a kid, because it's a day where you get gifts. Basically, the night before, on January 5th, you put your shoe beneath the Christmas tree, and then you wait, and the next morning, January 6th, you wake up excited, and you go to the tree, and you find toys or gifts or whatever the three wise men, which we call Tres Reyes Magos, they would give to you. So it's kind of like Santa for Christmas. So we also get gifts for Christmas, but this is the second time where we also get gifts for the three wise men. You have to put the shoe, because if you don't put the shoe, you don't get anything. which is why I didn't get anything because I didn't put my shoe this year. But anyway, so you put your shoe, you get your gifts, and then you go to school, and then you talk to your friends, and it is really exciting time. And later that day, you get together with your family to share the rosca de Reyes, which is called here King's Cake. And I know I recently learned that it is a tradition as well in the south of the US, like in Louisiana, they do King's Cake as well. So we do this rosca, we get together, we cut different pieces. And in fact, this photo is the rosca that I baked myself this year, which it looks better than it turned out, but at least it was something. And in this rosca, there are some plastic boys that represent Jesus. So if you cut the piece and you get the plastic boy, you need to organize and pay the party on February 2nd, when we again get together. So we never run out of excuses to get together with your family and celebrate. On February 2nd, we get together again, and then we share tamales and atole together. And that picture actually is a tamal that my brother cooked. So because we're both abroad, so we're not in Mexico, so we have to create our own food somehow. Anyway, so I just wanted to share this cool tradition that we do every year. And yeah, that's the end of my talk. Again, thank you to people who attended and to people that are working with me. And I'm happy to take any questions, but I will stop the recording first